This is SciFi Talks. Today in this episode, we would like to invite Dr. Neil Sheppard from Center for Cellular Immunotherapies. Since joining the Center for Cellular Immunotherapies in October 2019, Dr. Sheppard has brought invaluable expertise from his years in big pharma and biotech. At CCI, he heads the Think Lab, Therapeutic Innovation in Natural Killer Cells, and drives combination strategies for the clinical stage Mesothelin-specific CAR-T program. His work sits at the cutting edge of cancer immunotherapy, and we're excited to dive into it with him.
Welcome, sir. I think this episode is going to be very amazing because I think I can explore both the science and the business side of scientific project, mainly cellular therapy. So I think this is going to be a very amazing episode. Well,
yes, I'd be happy to talk to those. I've worked in industry for 12 years and I've been back in academia for about the last 5 years.
Is it possible to explain what you are currently working on and what are the projects you have in your lab and what does your lab do? So
I lead a lab at University of Pennsylvania Center for Cellular Immunotherapies called Think Lab, which is Therapeutic Innovation in Natural Killer Cells. So as suggested in the name, we mostly focus on natural killer cells, but I don't think you can be at UPenn without doing any T-cell work. And we do some CAR-T projects as well, of course. So the NK work in the lab is focusing on how to edit NK cells to sustain their activity in optimal ways. I think they're not being used correctly as of today. I think people have generally seen them as an alternate chassis. Perhaps it's easier to get into allogeneic space using the NK cells. And people have just taken a CAR from T cells and put it into an NK cell and expect it to work in the same kind of way. And I think the biology is quite distinct enough that is not the optimal way to use them. There's been several other barriers to NK cell research which have held them back. But I think if we can really understand the biology and including the disease biology where we want to use them, we can engineer them for optimal therapeutic applications. So that's what my lab is focused on. We currently focus mainly in cancer, but we're interested and have some plans to grow into other therapeutic areas in the next couple of years.
So we are already talking about engineering NK cells or engineering T cells, but we also have seen the examples of TILs where we don't have to engineer anything, just the TILs work without any engineering. So what do you think, how much engineering is required in the cells, whether it's T cells or in NK cells, to fight against the solid tumor epicenter?
Well, I think we're very lucky to be in an era of high efficiency gene and RNA editing. So the technologies have been around for a while, of course, the vector technologies where we can add transgenes to cells, but we don't have too much control of where the transgenes go. And then with CRISPR editing, that really made targeted gene editing a lot easier. There were several other technologies before that were very precise, but they required a lot of editor design to go after particular loci. So they weren't very high throughput. CRISPR was very high throughput, but it has drawbacks of doing a lot of off-target edits. And so now we see in CRISPR enzyme of improves fidelity with less off-target editing. We see new technologies like base editing and prime editing, which have far fewer kind of off-target edits, much better safety profile in terms of genotoxicity to take into patients, but also RNA editing with enzymes like As13D, which may allow us to avoid gene editing altogether and do everything at the RNA level. So multiplex editing is definitely here. So we'll be able to kind of deplete or knock out a multitude of genes as well as add certain therapeutic genes. And so I think that will be necessary in some circumstances where there's a lot of challenges, a lot of different checkpoints acting in tumors that suppress immune cells.
You were in the academia, you went to a startup or industry, and then you came back to academia again. So what motivated you to come back, or what attracted you to come back to the academia again?
And it's a simple fact that there's far more jobs in industry than there are academic positions. While there have been some new universities and some growth in departments of existing universities, the number of people pursuing secondary degrees has risen sharply. We've seen a huge number of people. It's basically become the default in many countries to go to university and obtain a bachelor's degree. And the default used to be just getting that diploma. So to differentiate themselves, many people go and do additional degrees, as I did. But then I was very interested in academia anyway. In industry anyway. I had a year in industry as part of my undergraduate degree, but gave it a taste of it. It's part of a natural flow because there are, I think, that every job in academia will be 9 or 10 jobs in industry. And I was quite interested in applied science. I wanted to learn more about the translational and early clinical space, which is very tough to do in academia, and except in a few institutions. So that's why I moved into industry after my PhD, and very much enjoyed industry, went through various companies and positions. But when I moved into cell therapy space in 2015 full time. So I'd been in industry for about 6 or 7 years by then, mostly in the vaccines field. And I moved over to cell therapy. And what I realized in a space of a few years is that this is definitely not a mature technology. And there are fundamental paradigm shifts coming, which actually make it a fairly poorly suited time to be in industry in that sector, because a lot of assumptions do not stand the test of time, and including these new medicines. The first CD19 CAR-T therapies were approved in 2017. I think it's likely that they will be replaced with new versions. Certainly we see people like Novartis looking to introduce a 2-day cell process down from a 9-day cell process. But now, a colleague of mine at Penn has published a 1-day cell process. So the cell therapy manufacturing is being completely revolutionized. And I think a lot of industry have put large capital investments in manufacturing facilities that may be completely outdated within 5 years, which is not the kind of investment time horizon you say. If you build a manufacturing facility to make a monoclonal antibody, probably you can still use the same facility in 10 years without major modifications. I don't think that that's true for cell therapies because things are changing so much. And even the question of will cell therapy become allogeneic or not, that was a big assumption that after autologous therapy was successful, that allogeneic therapy would replace it, but it hasn't happened. And most allogeneic therapies have shown poor efficacy and poor PK, and that they're quite rapidly eliminated. So that science is still really being driven by academia. So I had this wonderful opportunity to come to a space where it's kind of like a biotech in academia, because we have our own GMP manufacturing facility for vectors and cells. We have a clinical trials unit, so we can do everything from basic science in the lab through to clinical trials, including combination clinical trials. And unlike in industry, which I've also experienced, if you're in a biotech company and your lead asset is cancelled, most people lose their jobs, and the biotech company is usually sunk within a few months if that's happening. Academic already have 1 or 2 other candidates waiting in the wings, waiting to go in based on various hypotheses and data-driven hypotheses of why the first generation wasn't good enough. But in industry, you place a large bet on your lead asset, and you rarely have the cash or the time to recover from the loss of a lead asset and go back. So, I've seen companies build and invest in very expensive manufacturing suites and then have to sell them and lay off everyone because their main asset failed. So that kind of is the last kind of 5 years or so. Cell therapy is not a mature technology. It's rapidly evolving. And so it's difficult for industry to be in the space unless financial deep pockets and long-term time horizon commitments that perhaps some of the bigger pharma companies like Johnson & Johnson have.
Often we don't talk about the manufacturing in the academia. And I think it's like your manufacturing is more related to the industry. And do you think that, I don't know if I'm right, but do you think there is not such an open collaboration between the industry and academia when it is about the manufacturing and this kind of downstream research?
I think it's quite the opposite, actually. So in the first question, we in academia manufacture our own products for our own clinical trials, and in industry, they will manufacture their own products for their own clinical trials. In industry, you have to meet a much more rigorous standard. We generally all have to meet GMP, but in industry, if you're preparing for a commercial launch, that's an entirely different scale. something that we would never go near or worry about that too much in academia. But actually, I see time and again, it's, yes, industry can be inventive, but most of the inventions happen in academia. There was a paper, I think, in around 2019 or 2020 in Nature, looking at where the IP for cell therapy sat, and they had the top 10 institutions and the top 10 individuals in the world. And my current institution, UPenn, I mean, I wasn't part of it at that time, but my current institution was the top and had multiple scientists in the top 10, and also had more than twice the number of patents as the next company, which next number 2 on the list, which was Bristol Myers. I think industry takes well-qualified scientists, but then it usually shifts them from therapy area to therapy area, and they cannot be at the cutting edge of cell therapy unless that has been where they did their PhD and where they did their postdoc. If they did it in anything else, they're not going to be at the cutting edge, and they may make strident efforts, but it's not the industry that published the one-day cell process.
Yeah.
But it come from academia. And even now, the thought of in vivo CAR-T therapy, so another UPenn spinout, and I think there are a handful now, maybe 3 or 4, doing in vivo CAR-Ts, either with vectors or with lipid nanoparticles. But those are all recent spinouts from academia. So they come with a lot of academic IP, and then because it's towards commercial intent, they need to spin out into the biotech space and raise the capital and do the trials. But it's that has all come from academia, and big pharma basically don't do that much innovation in-house. They acquire innovation. Big pharma is excellent at developing. They have the best clinical development, the best kind of marketing, regulatory in the world, but they acquire innovation and they're professional at it. And fully develop, commercialize it. But having worked in all of those settings, so biotech and big pharma and academia, I think the pace of innovation in academia is just so much faster.
Again, I would like to go back to the engineering. So there are several multiplex tools or high throughput tools, like which can give the high throughput data. So there are so many tools there, but do you think they are enough to do another next level of engineering to create a better engineered cells, or they are not enough, or are we waiting for other technologies?
So methods are only tools, and tools can be useful, but they can also be misleading and misused. So I would never want to rely on a single tool. And I think if you look at the way drug development, drug discovery works, there's always a big element of luck. And again, a lot of it comes from clinicians, physicians, scientists, and scientists who are very close to the disease area and who spot new things. So, and there's a great deal of luck that makes a difference, and timing that can make the difference between a successful and an unsuccessful. So yes, it's really good to be able to do multiplex efforts and to be able to do high throughput screening, but the companies that do high throughput screening the best are the large drug companies. And they've been telling us for years, we've got millions of compounds and we can screen, we'll set up an assay, we'll screen millions of compounds. We've got a target, we'll make dozens of antibodies, we'll test all the antibodies. But still, they end up buying biotech and academic antibodies and drugs and cells because they weren't looking at the right target, or they weren't looking at the right piece of biology in their high throughput system. So you don't need high throughput to succeed. It can help when it's used well in the correct setting, but it's certainly not a solution in of itself. Otherwise, innovation would have been completely overtaken by big pharma, like from the 80s when they set up certainly high throughput small molecule compound screening was professionalized in the 80s and 90s. And screening hundreds of thousands, if not millions of compounds against different enzyme targets. And yes, they found some. They did have some hits, but still most of the drugs that are on the market came from small biotechs. They acquired perhaps a different approach.
Why cellular therapy is challenging is it's because of it has several challenges. So there are several challenges like related to T-cell infiltration or homing or on target of tumor, these kind of challenges there are. But so as a researcher, how do you choose the right problem? Because you have to invest a lot of time tackling that challenge or solving that challenge. So how do you choose the right problem as a researcher?
That's a really excellent question, because in terms of drug development, you have to solve all of the problems and you have identified the correct patient demographic. Otherwise, you'll never have a successful drug. including a cell therapy. So every drug has to have acceptable safety profile. So if your cell therapy is very efficacious but very toxic, it will never progress beyond phase 1 clinical trials if that makes it that far. If it's very safe but not efficacious, it will also not progress beyond phase 1 clinical trials if it makes it that far. You have to solve all of the problems, and that's very difficult to do, but a lot of the problems can be interrelated. So in cytokine release syndrome and neurotoxicity, for example, with CD19 CAR therapies, it's to do with the ability of T cells to make a great deal of cytokines, and sometimes that can be an overproduction and stimulate other cells in the body that kind of lead to a cascade. And it's also related to the antigen burden, so how much disease a patient has. And what was quite nicely shown with natural killer cells is a CD19 CAR NK had no cytokine release syndrome and no neurotoxicity, because NK cells don't expand in the same way as T cells do. They do expand, but not to the same extent. Now, the biology of a T cell, the natural biology of a T cell is that you have just one or a small handful of T cells in your body that can recognize a specific antigen, and you've got to find them, and you've got to make a whole army out of them, hundreds of millions of billions of them. So T cells are predestined to expand logarithmically when they encounter their antigens, whereas natural killer cells are much less specialized. They are specialized, but much less. They're not antigen specific, and they're kind of produced by the bone marrow at a more or less constant rate. And once they're released from the bone marrow, they don't really proliferate that much. And they have a short life expectancy. Their half-life is only around 10 days or so, 10 to 14 days. So they also have a different cytokine profile. So the CAR NK cells didn't produce the toxicities, but still managed to achieve the efficacy in the CD19 CAR setting. So that was pretty promising. Because it's an off-the-shelf product, it could potentially be cheaper as well, and it could kind of get out of the tertiary medical centers into community infusion centers, perhaps. So that kind of therapy potentially solved multiple issues at once. It still had some issues of its own. But yeah, it's difficult to make that kind of stride, because it's much more natural to try and fix one problem at a time. And then you get to the clinic and realize that one problem wasn't sufficient. And certainly when it comes to immune checkpoints, we've had problem of understanding which are the dominant checkpoints. And you've seen really the whole of big pharma that are involved in immunotherapy. After PD-1, the big success of PD-1 and PD-L1, they have tried virtually every other checkpoint that can be found, right, with blocking antibodies. And as of today, what has been approved is a LAG-3 antibody, but only in combination with PD-1 and only in certain melanoma patients. And then a couple of CTLA-4 antibodies in very restricted conditions. Again, I think mostly melanoma patients. Everything else has drawn a blank. So multiple attempts to block TIGIT and CD96 and TIM3, LAG-3 and other indications, VISTA, all of these. Not to say that they're a bad choice, but they just haven't stacked up. Perhaps we don't understand the relative importance of the checkpoint, but then there's been some really great data from groups like John Werry's group that show that PD-1 is a master checkpoint. And that's probably why PD-1 blockade makes such a difference. But of course, in the real world, only a minority of patients respond to PD-1 blockade. So yes, for the 20 or 30% that respond, they can have some really magnificent responses. But you still have many patients who don't respond or only respond for a short time. So it's a really great question, because we haven't solved it. And that's why you see, it is a little bit like a Vegas casino. Companies develop data packages of why they believe in a certain checkpoint or a certain target. But as we all tend to do confirmatory research rather than disconfirmatory research. So that is which our biases make us look for data that will support our hypothesis. But in the real world, doesn't care about our hypothesis. And, we often, unfortunately, get clinical failures when we get into the real world.
There is also another confusion in choosing the cell type. The T cells were previously the T cells were more popular, but nowadays, still they are popular, but nowadays the NK cells and the macrophage are also being used. So how do we choose the best cell type? And also, there is also confusion when choosing the source of the cells, the primary cells, like whether to use the primary cells or cell lines. There are several confusion in this respect also. So how to solve this kind of thing?
I think that's another excellent question. And I think even in the microcosm of Penn, there have been spin-out companies working on CAR macrophages, CAR-T cells of various kinds, and dendritic cells. And perhaps in the future, maybe NK cells, there are plenty of NK cell companies out there too. There are companies that work on gamma delta T cells, natural killer T cells. Pretty much every immune cell has been looked at, and many of them have been advanced into the clinic by this point. But they all potentially could have a setting where they could be an ideal cell type. I think it's really important that just because you have a hammer, every problem is non-nail. So you have to have a right tool for the job, and we don't always know what the right tool is. But the immune system uses all of these cells. Most of these cells are essential, or nearly essential to survival, right? So they all have a job to play, and some of them may have more. Some cell types have been described as architects of the immune system, or conductors of the immune system. So more likely to be able to foster an immune response in the patient, which especially when it gets to cell therapy and to solid tumors, because there's a lot of antigenic diversity in solid tumors within and between patients. You really want to try and break immune tolerance against the patient's own tumor. It's going to be very hard or I think impossible, to tackle a solid tumor in a reliable way with a single antigen specific CAR-T or TCR-T cell, because all the tumor has to do is lose the antigen. And most antigens that are targeted are not essential, so the tumor can continue without them. So you ultimately want to be able to induce an immune response. And dendritic cells are clearly architects of the immune system. They're foundational to inducing immune response. I'm not sure really how they will perform as cell therapies, but the first approved therapy in cell therapy was a dendritic cell-based Provenge for prostate cancer. So they can work for sure. I think NK cells have been described as an architect of the immune system because they're very good at calling in the host's dendritic cells. And that has actually been shown to be a key feature of NK cells. But again, when it gets to the real world, all those potential sources of bias fall away. So the proof will be in the trials when they happen. But it's also important to make sure that you have the most robust cell therapy, that you're developing in the right condition, the right setting. You can certainly have a promising cell therapy, but then do the wrong trial and show no benefit. So unfortunately, again, you have to get multiple things right to get a good signal. And many times we don't get a good signal because we didn't get everything right.
So you think that there would be always the confusion in terms of the cell type, which cell type to choose, and this kind?
I think in certain areas we know, obviously CD19 CAR therapy and now BCMA CAR therapy has been very efficacious for B-cell malignancies. We think in these indications that many of these B-cell malignancies have very little antigenic diversity, but they're all, or very nearly all, labeled by CD19 or BCMA, although in both cases we see antigen escape. And so I think in the future we'll be targeting more antigens. So people are looking at CD20 and 22, or in myeloma, they're looking at GPCR5D and potentially other antigens to try and prevent antigen escape. T cells seem to be the perfect cell there, with the drawback of the toxicity. So we have to find a way to deal with their toxicity. Perhaps that could be done by NK cells. I'm not sure. I think NK cells may be better suited to other paradigms, other treatments, and in certain combinations. I think we see some on the cutting edge with the myeloid cells right now. Perhaps macrophages as a therapy haven't really shown much of a signal yet, but it's still early days. But there's a movement, I think, with lipid nanoparticle technology, and people like Moderna are involved, to instead do monocytes, because monocytes, are in the blood, but then enter the tissues and turn into macrophages. And that may be a good way to get your cell therapy to where it needs to go. So certainly, I think each of the cells could have some value. And we've seen, obviously a lot of positive data for T cells in heme malignancies, and a small amount of positive data in solid tumors with select indications. We've seen some promising data for natural killer T cells, but natural killer T cells are much more similar to T cells than they are to NK cells. So I think that needs to be borne in mind. I think some of the early data with gamma delta cells are interesting, but again, here there's a lot of difference in the field, because some people's approach is basically to grow gamma delta cells, to isolate them and expand them. That's can be done, but it's quite difficult. So an alternative approach is just to take the gamma delta TCR and express it in an alpha beta T cell. And that actually seems to work really well and allows you to use the standard T cell production process that is used for alpha beta cells, such as in the CAR-T cells. So that's kind of one to watch, and it'll be interesting to see what is the right way to use the gamma delta TCR. Is it in a gamma delta T cell, or is it in taking it out and put it to an alpha beta T cell?
Just one question, like, is the CAR or the artificial receptor that we are using, is it perfect, or do we need to engineer it a little bit more to make it more better, or is it perfect for now?
It really depends. So certainly there's lots of data out there that for NK cells, you can pretty much take any CAR and put it in a T cell or an NK cell and it will work. But it's not necessarily what's optimal for T cells is not necessarily optimal for NK cells that have a number of different active signaling pathways present. However, I think with NK cells, we actually also need to consider whether we are overstimulating them, because they have a lot of different, their biology is to integrate multiple signals from different sources, less than to have one huge on signal. And I think because natural killer cells recognize tumor cells anyway, that there is quite a lot of opportunity to enhance the natural recognition rather than to necessarily, I think one of the big mistakes that's made currently with NK cells is the concept that you just drop in a CAR and you're treating them like a T cell, but they're not a T cell. And yes, you can get a cytotoxic cell that way, but it may not, we're not yet to see a lot of really positive data in that field. I think we do better. We need to understand the NK checkpoints better, and we need to understand how to basically sustain the NK activity, or how to dose the NK cells for maximum activity.
To play with surface antigen, the artificial receptors are perfect. They are the upstream factors, but if we there are also the changes in the downstream players, like transcription factors, microRNAs. So how do you choose, or how do you know that which factors are more important to make efficient CAR-T cells or efficient CAR immune cell?
So I think each immune cell will have its own answer there, but there's not necessarily one correct answer. I mean, my lab is involved, the labs are doing multiple different approaches. So applying small molecule compounds to the culture medium when you're growing the cells can be quite useful. So you can change the nutrient mix, you can add certain, there's been some publications quite a while back now about adding epigenetic inhibitors of the enzymes that modify the epigenome. Things like that you can add to culture additives that actually lead your cell to have quite a different type of activity when it's come off the production run. But we need to understand how long that lasts, and is worth the squeeze. I think it's very promising to look at things like knocking out or overexpressing certain transcription factors permanently or temporarily, because that can have a big effect as well. It's possible that we've certainly seen some evidence that perhaps CAR-T cells in solid tumors settings, they could be converted into CAR-Tregs, and they may want to prevent that happening if that's part of a disease escape. So there's quite a lot of exciting things that can be done out there, and there have been some good publications out about overexpressing signaling pathway molecules like c-Gen from Crystal McCall's group. That's an excellent, and I think it's in the clinic now, really made the CAR-T cells more potent, knocking out other, we had a publication about knocking out some factors called regnase and roquin, which are natural inhibitors of inflammatory responses, and that led the T cells to be much more robust. There's a company advancing tumor infiltrating lymphocytes into the clinic now that have had a protein called SOX1, suppressor of cytokine signaling 1, knocked out. And the data about has been around for a while that knocking out SOX1 improves the kind of the activity of the cell, but potentially also its metabolism. And these aspects of the biology are often interrelated. It's very hard to change one thing without changing another thing in the cell. And that's where, we often think about, and in publications, people refer to kind of biology as being like hackable and being able to write code, because we can make mRNA scripts and everything. And yes, that's true, but only to an extent. And one example of that is even the work that was done several years ago with CAR structures, where the first generation of CARs had CD3 zeta signaling or FC receptor signaling only. And then those weren't efficacious, although all the trials were in solid tumors. They didn't go anywhere. They were replaced with the second generation CARs that had CD28 or 4-1BB signaling. And that's when we saw really great clinical responses in B-cell malignancies. And so the kind of next step was, we'll just put multiple co-stimulatory domains in. We'll have CD28 and 4-1BB. But you have to remember that the way these things work is that physical location of signaling molecules is also really important. And it's basically been found that, yes, you can add multiple co-stimulatory domains, but the one nearest the membrane, nearest the interleaflet membrane, has the dominant effect because it is in the correct location where naturally it would be to access the membrane proximal signaling cascades. The further away into the cytosol you have it, the less active it seems to be. So I'd actually not seen, I don't think any of those so-called third generation CARs that had combined BB and 28 signaling or ICOS or anything else have really gone forward at this point. And that's because that position was really important. So it had the dominant effect. But another kind of explanation of that is we've seen, going back to the cell therapy manufacturing that I talked about earlier, the first cell therapy manufacturings for CAR-T cells and TCR-T cells, the process is usually 9 to 11 days. We know now that shorter processes are better. So the 3 days, the 5 days is better. 3 days is better. 3 days is better. One day, excellent. And unfortunately, quite a lot of the work that has gone into so-called fixing CAR-T cells is fixing a problem that has arisen because of the kind of suboptimal way we've been making them. So, if you have a 9-day process, your CAR-T cell is naturally going to have less naive cells. It's going to be more differentiated. And so it's going to have certain problems that maybe you can fix. But if you just had a 3-day or a 1-day cell process, you wouldn't have those problems in the first place. So you might not need to fix them. So this has added a lot of extra complexity that cells as drugs doesn't exist in the same way with small molecules or biologics as drugs.
You are already in that academic environment, which can generate a lot of cell therapy based startups. So there is a competition. I think the cell therapy startups are already crowded. And what do you think? So what's your prediction? And what do you think? Who wins the race at the end? So because there is already a competition and a crowd, and competition in terms of technology, competition in terms of team. So who wins the race at the end?
That's an excellent question. And I guess I also like to start off by saying that cell therapies don't necessarily have the right to exist. So I think bispecifics, like we see bispecifics against BCMA and other antigens, they're making a lot of progress. They've greatly improved the PK from what the first bispecific blinatumomab had such poor PK, it had to be administered by a constant infusion pump. Gone are those days, basically. Now we're in a much better place with bispecifics, and in the treatment of myeloma, it's only early phase, but there was a combination trial of 2 bispecifics that had a 95% response rate. That's excellent. That's really very strong. Of course, the CAR-T cell-to-cell had a 100% response rate, and most of the patients are in response 1, 2, 3 years later. So cell therapies can be very potent, but they don't naturally have a right to exist. We will go where the technology goes. And I think in the cancer field space right now, it's very exciting to see the neoepitope vaccines, which I think are going to be much more successful in kind of minimal residual disease setting where you can catch the patient early enough to surgically resect their cancer, and then you're kind of immunizing them to try and stop it coming back. But there are other agents in that space as well. So the study of an oncolytic virus that was given in patients with borderline resectable pancreatic cancer. So there's very likely some disease left. And it was injected, and that seems to have done the same kind of job as a vaccine. So those patients had much better survival, much less recurrence. So those are really exciting developments. I mean, I think cells can be extremely powerful, but we have the tension between autologous, where we're moving the field gradually from a 9 to 11 day process down to maybe a 1-day process for T cells, and maybe moving it to the bedside to do local manufacturing. That is up against allogeneic, which I think is a risk to allogeneic because allogeneic, you have to make very large batches of cells to treat hundreds of patients. And we know that the more you grow cells, the worse they become. So I think that's always kind of a limitation for some of the allogeneic therapies. And then in vivo. So can we just replace CAR-T cells with in vivo therapies? But it's nearly always the case that the one that goes first shows the way. So CD19 CAR is very successful. So it's not a surprise that the in vivo CAR approaches are mostly targeting CD19. Because it's a clear market that you can take, and maybe you can do it with more safety and far lower costs, much more convenience. So I'm excited about the future of in vivo cell therapy. It's not necessarily straightforward. Until we see how well these things translate into the clinic, we have to have hope. And some of the primate studies have been promising. And primate studies usually translate well, but the old saying is that mice lie and monkeys exaggerate. So let's wait and see. So it could be that autologous cell therapies are here for quite a while. I think it could even be that lentiviral vectors are here for a while because they just work well, and we've got the costs down to some degree now. So how to win the cell therapy race? Yes, if you can get cheaper, safer cell therapy, that would be great. I think we have to find ways to treat patients with these. 90% of the tumor need solid tumors on malignancy. So there's definitely some heme malignancies that are yet to be treated, and actually there's some early exciting data in T-cell lymphomas, which are much rarer. So I hope if we look out, say, 10 years, I expect that most hematological malignancies will have an approved cell therapy. However, there may be a lot of competition with bispecific agents and things like that. And I think we will see more than just T cells approved. We'll see some of the others approved. But yeah, cell therapies don't necessarily have a right to exist. So as new technologies come along, they may go away. And actually, I think in vivo technology is potentially one of those technologies. And again, I'm obviously a bit biased towards NK cells because I work on them, but in general, NK cells work better in the allogeneic space because they are inhibited by your own HLA. So if you use them in somebody else, one of those major sources of inhibition is removed. And that's something that you can look at the degree of match, and you could potentially design a product to maximize mismatch, or you could do gene editing to create universal products that will be more efficacious than being able to modify the patient's own NK cells.
Is there a challenge of expanding the NK cells in the lab?
So in the factory, in vitro, we can expand NK cells millions of fold. So we have the technology to expand NK cells, and the NK cells that we expand are apparently efficacious in animal models and as efficacious in vitro. Whether they are what we need to understand better is it going to follow the T cell paradigm, where the more you expand a cell, the less efficacious it becomes. So that's actually something quite interesting to look at, which actually disfavors allogeneic cell therapies, because in general, you have to expand allogeneic cell therapies more in order to give them to multiple patients. In vivo expansion is an issue, but I think it depends on what you're using them for. So I've heard this said as a way to describe an NK cell therapy in the future. It could be a very safe form of chemotherapy. So a lot of chemotherapy drugs are still used. They're horribly toxic as their mechanism of action to kill all divided cells. The hope is that there's a therapeutic window there, and you can damage fewer stem cells than cancer cells. But they have horrible side effects. They make people feel terrible. Their hair falls out. All of the other nasty things that happen to patients on chemotherapy. But NK cells, yes, they don't persist, but maybe you can infuse them every week. There you could go to the clinic and you could get repeat infusions of NK cells, and the NK cells are engineered for optimal activity against the patient's tumor. You could perhaps get activity with much less toxicity. It may work very well in certain indications or with certain combinations. So you might not need big expansion or persistence. You need some degree of expansion and persistence. And so it does make sense to work there, but I'm not one of the scientists who is trying to turn a T cell, an NK cell into a T cell and have it survive for months or years, because I think that goes too strongly against NK cells biology. And actually, there's an element of safety and a lack of persistence.
What is your view on using stem cell lines in cellular immunotherapy?
So I think it definitely is exciting conceptually, being able to do all of your gene edits and then select the perfect stem cell that has everything you want, nothing that you don't want. and potentially grow it indefinitely, and make large amounts of NK cells. There have not been any head-to-head trials because we're only at the phase of phase 1 trials, basically. But so far in the NK field, the best data have come from groups that are using primary NK cells. So that's either cord blood NK cells or adult healthy donor NK cells. I question with the iPS leak how much like a real NK cell it truly is, because NK cells have to undergo a process called education as they develop in your body, where they learn what to kill and what not to kill, and that requires interaction with your HLA molecules, and it requires interaction with multiple cell types. That doesn't happen for an iPSC-derived cells, and I think that's a big black box. We don't know what is happening there. They do encounter each other, but they don't encounter anything else. And if they've been engineered to be HLA negative, how educated can they be? So what we see with poorly educated NK cells is that they either lack function altogether or they rapidly lose their function. The other aspect I'm concerned about iPSC is that they're clonal, which is different. So depending on how many markers you look at, your own NK cells are very far from clonal. So there are thousands of different subpopulations. And some may be better than others for different tasks, but a iPSC-derived NK cell will be clonal, which means if it breaks immune tolerance, especially to antibodies, everything will be wiped out. So you won't be able to redose with anything from that clone. Whereas with NK cells, you could potentially, you could make a batches of therapeutic cells, say from 6 or 12 different donors. And even if you wanted to give the patient a long course of therapy, you could perhaps change the donor every 1 or 2 weeks, every 1 or 2 cell from the same donor, and try and sidestep the the clearance response. That's been one of the problems holding back allogeneic therapies, is that they're immunogenic still. Despite lots of engineering, they're still pretty immunogenic and they get wiped out. So that will need to improve as well. And there has been some promising data. I think Fate Therapeutics had some data, but when the Fate Therapeutics crested, I think, at almost $130 a share based on their off-the-shelf activity in several indications, and then after 6 months, all the responses went away, and they went down to, I don't know what they're at now, like $2.50. So it's really difficult. Maybe they're not using them in the right setting, but that is a setting where T cells are the best. And kind of one thing that emerged from the CD19 CAR NK trial, which was very promising, and those use cord blood cells, and they could be detected 1 year later, which was actually really unexpected because they're allogeneic cells, and there was no specific immune stealthing. So they didn't have their HLA deleted, they didn't have overexpression of any immune inhibitors, but they could still be detected at low levels a year later. So that poses some interesting possibilities. Cord blood cells, they have some very interesting biology when it comes to their HLA expression, but may explain why they're able to persist so long.
Do you think that microfluidics or 3D organoids, spheroids, this kind of models are contributing enough to the cellular immunotherapy field, and how they are contributing?
And there are multiple different assays and systems under each umbrella. So, microfluidics, for example, are used to make lipid nanoparticles. Microfluidics are used to do kind of organ on a chip. And they're used actually to create single cell suspensions for single cell analysis. So they definitely have multiple applications that are really helping us get some quality data. I think the organoids, organs on a chip, this is one area my lab is going into as well. I think, so much work is done with 2D cultured tumor cells. And we know that is quite different to a 3D culture, even a basic 3D culture. So in the right culture vessel, essentially every solid tumor line will form a spheroid instead of a flat monolayer. And the biology of the spheroid cells is different. And we don't understand enough about how different it is, but it's probably more like a real tumor. And as it grows, it does tend to get a hypoxic core, and the cells at the center eventually die. But there's a possibility to use spheroids that are composed of multiple different cell types. If you can get those cell types to sit together well, there's a possibility to derive them from patient tumors directly or from patient-derived cell lines to try and make something that is much more representative of real biology. That's one of the things I see that basically every lab has to do is, for example, you want to target HER2 with a CAR-T cell, so you go and look for HER2 positive tumor lines, or you engineer your tumor line to be HER2 positive. But in the real world, even if you have a HER2 positive tumor, there'll be a lot of cells in that tumor that are HER2 negative. So, and I certainly have a colleague who's done some experiments in injecting 95% positive and 5% antigen negative tumors, and CAR-T cells cannot control the antigen negative tumor. It just doesn't happen. So that also helps understand why it's been so difficult to treat solid tumors with CAR-T cells so far.
I think we are almost in the end of the episode. I would like to know about the future of cellular immunotherapy. What do you think about the future of cellular immunotherapy, and how do you predict it, and what kind of technologies can we see in next few years?
That's an excellent question. So, my opinion may change if in vivo cell therapy works out very well, as many people hope that it will, and as it has done in some animal models. I think autologous therapy is probably here to stay, at least for a good while, and that will be based on much shorter manufacturing that leads to a much more potent product, and potentially manufacturing in the hospital setting. So without all the logistics involved in shipping and flooring and all this stuff. Allogeneic cells, I think, will find some space eventually. We will see multiple cell types developed, potentially as combinations. That's kind of interesting. And that's actually one interesting aspect of iPSC. You could potentially turn the iPSC into any cell. So you could have a combination that is alpha beta T cells and gamma delta T cells and NK or KT. So we will see more of these cell types move forward in clinical trials, and I think eventually be a product. I think we will deal with most, if not all, heme malignancies, and I think we will start dealing with some of the solid tumors. That's going to take a lot more engineering, but I think we will get there. And I think other things that we need to do is, obviously find ways to reduce the costs and the toxicity, so that more patients can benefit, but also potentially eliminate lymphodepletion by using other strategies and combinations. The lymphodepletion is almost universally used with cell therapy. It's often Cytoxan and fludarabine, but there are other regimens, and it has a lot of toxicities. And unfortunately, some patients have severe toxicities as a result. Some patients develop neutropenic sepsis, and unfortunately, some will pass away because their infection can't be controlled. Some patients will have bone marrow failure. So it's something that we would like to eliminate, I think, in the future if we can find other strategies.
Thank you so much for the insightful discussion. Thank you so much for the conversation, sir. Thank you.